Good morning, viewers. Welcome to MouShauk. Uh, with, in my last video, I ended up with the small intestine villi part. Okay, so that is the ileum where we have lots of finger-like projections to increase the surface area of absorption. That is, those are the villi. Okay, so we will start with that. Uh, Actually, the diagram which I told you, I will produce you today. I mean, in this video that I have made, and we will also discuss the rest of the small intestine part. Okay? So, before I start, I would request you to please to subscribe my channel if you have not subscribed yet, and if you are a new viewer to my channel. Okay. And if you are liking the video, press the like button, share with your friends so that they can also be benefited from this video, and do meditate before you start your day. Okay. So let us start with the meditation, short meditation. For that, please concentrate on a point of light. Get the light in the bottom of the screen. The very bottom. It's light with light. Light with feeling the light with problems, the light with tensions, the light with the outside of the bottom. And you can try like this. And invite the sky very happily, feel like a baby you're flying, going down to the sky, there's a really very, which is very warm, there's anything that you need. Your feet are very well, so light and light, without any burden of you. The gratitude, that you can fulfill all your wishes. Powerful energies on your side, supporting you to every step. Which is happening, no one has been saying it's wrong. It will come back to God, it will be body, your own body will returning to it. You are thank you God for being with you, and you can see that you're getting appreciation with regards to clearly you are free of all body. And you're feeling light without any problems. It's impossible. Yes. So let us end here and start with the discussion. Okay. So for the discussion, see, this is the picture of a villi. Okay. See, I told you the villi has a single line of epithelium, the finger-like projection with a single line of epithelium, and also it has capillaries. One is of artery, another is of vein. Okay, and both are interconnected. Okay, and in between the blood vessels, there is the lymph vessel, which is known as lacteal. Okay, and so this is the red one is the artery, and the blue one is the vein, and the capillaries are in between, interconnecting both the artery and the vein. Okay, and this blood flows through the artery, moving to the veins, and then after that, the vein is connected to the hepatic portal vein, that is leading to the liver. Okay, the lymphatic duct carries the substances to the lacteal. I mean, from the lacteal, it goes to the lymphatic duct to the different parts of the body, and again, finally it will mix with the bloodstream. Okay, this is the microscopic structure of the intestinal villus. Okay, and between the two villas, the another one will be another the side and the side, the two villi. So in between the two villi, there are glandular cells which secrete what? Intestinal juices. Okay? Now see, I told you that the intestine receives three types of juices. One is from liver, that is gallbladder, through gallbladder when it is stored there, and the liver releases the liver releases this juice, and then it is stored in the gallbladder, and when it is required, they will release it. The gallbladder will release it to the common bile duct into the duodenum. So this is bile, which is secreted. One is bile. Another one is pancreatic juice from the pancreas. Another one is the intestinal juice from the glandular cells present in between the villi. Okay. So first, let us move on to bile. What is bile? Bile is a yellowish, green, and watery fluid produced in it. It's a yellowish, green, watery fluid that is produced in the liver. Once when we vomit a lot, when nothing is there in the stomach, when we we're going on vomiting, then you can see your mouth turns bitter. Okay, and some fishy smell comes in your mouth, and the color of the vomit which comes out, it is slightly greenish water. So that is what that is why it comes out. Okay? Then they are transported through the hepatic duct. Okay? After joining with the cystic duct from the gallbladder, hepatic duct from the liver. Okay, so hepatic duct carries the bile from the liver to the and joins to the common bile duct. Okay, and cystic duct from the gallbladder, where the bile is stored in gallbladder, where the bile is stored. There, from there, the cystic duct carries the bile to the and joins with the common bile duct. Okay, so and they open directly into the duodenum. This common bile duct opens directly into the duodenum. Then they may flow directly into the duodenum, or they can be temporarily stored in the gallbladder, and when required, it will flow into the duodenum. Now, why this color of this bile color is yellowish green? Okay, it is because the pigments, there are two pigments which break down, okay, of the when the RBC worn out RBC, when an RBC gets worn out, it has two pigments, okay, bilirubin and biliverdin. Now these two pigments also breaks down. These two pigments also break down, and due to this broken down bilirubin and biliverdin, the color of the bile becomes yellowish green. Okay. Now, this bile contains a lot of sodium bicarbonate or sodium hydrogen carbonate that neutralizes the acid content of the food. We know that when the food which is coming out of the stomach, that contains a lot of acid, because the stomach juice which has come out, that has HCl acid. Okay, HCl hydrochloric acid it has. So the stomach, when it is when the food is in the stomach, it will mix with the HCl, and that food will be coming out from the stomach little by little into the duodenum, fall into the duodenum. Now here the food will get the bile juice, which will make it alkaline. Lot of sodium carbonate is there. It will neutralize the food. Okay, the sodium bicarbonate is an alkali, so it will react with the hydrogen chloride and hydrochloric acid, and it will neutralize the food. Okay, and it will make the food what? It will make the food alkaline, so that the pancreatic and intestinal juice, which contains the enzymes that can act on it. So why do we need the food to be alkaline? Due to so that the pancreatic and intestinal enzymes can act on this food. Okay, so to enable the food to act with the enzymatic action, that is to make the food act with the enzymes, okay, of the pancreas and the intestinal juice. So the food has to be made to alkaline and not acidic. So the acid has to be neutralized. This is neutralized by sodium hydrogen carbon, which is present in bile. Okay. Now the bile salts reduce the surface tension of the fats. Another function of bile is they will reduce the bile salts. They have some salts also, so that will reduce the surface tension of the fat, so that the fat droplets can be broken down into simpler fat, tiny droplets. Okay, the fat droplets can be broken down into tiny droplets, which is known, this process of breaking down of fat into tiny droplets is known as emulsification of fat. Okay, so that it provides greater surface area for the enzymes to act on it, and the fat can be digested. Okay, so the fat to get digested, the fat substances, the fat molecules have to be broken down into tiny droplets. Okay, this is done by bile salts. Okay, that is, emulsification of fat is done by the bile salt. Okay, so in a nutshell, what do we see? The substance, the food substance which is coming out of the stomach, that is known as acid chyme. Now, this acid chyme gets the bile on it in the duodenum. When it is coming out into the duodenum, the bile acts on this acid chyme to make it alkaline chyme. Okay, same chyme is there, but it is from its acidic nature will change into alkaline. Okay, this is due to the sodium hydrogen carbonate present in bile. Okay, now the fat. Okay, fat also will get emulsified due to this bile. Okay, so now the food substance which is present in the duodenum is now alkaline in nature. Now what happens? Now the pancreatic juice acts on it. It is now this alkaline chyme is now fit for the pancreatic juice to be acted on it. Okay. Now this pancreatic juice is the juice which is secreted by pancreas. What is pancreas? It is a whitish gland which is located in the stomach. It's a whitish gland which is located, behind the stomach, not in the stomach. It is behind the stomach. It is leaf-like structure. Okay, it will be if you see the picture of a pancreas, it will be a leaf-like structure which is behind the stomach. Okay. Now the pancreatic duct opens into the duodenum by an aperture which is common to that of the bile duct. So the aperture of the common bile duct and the pancreatic duct is the same. Okay, the same aperture is used for the which is present on the duodenum for the common bile duct and the pancreatic duct. Okay, this pancreatic juice contains three kinds of enzymes: amylopsin, trypsin, and strepsin. Okay, amylopsin, trypsin, and strepsin. Now, in a nutshell, we can see this pancreatic enzymes, how do they, on which food they are acting. Now, the amylopsin, it's a pancreatic amylase, which is acting on the leftover starch. We know that starch digestion takes place in the mouth, where the salivary amylase acts on it. Okay. Now, the leftover starch, which has been passed from the mouth to the duodenum via the esophagus and stomach, that will be digested here, where amylopsin or pancreatic amylase will act on this leftover starch to change it to maltose. Okay, it's a simple maltose. It will change into maltose. Now, this Again, enterokinase. Enterokinase is another enzyme which is present uh, in this uh, place. Okay, it will change into inactive trypsinogen to trypsin. Okay, so the trypsinogen first, the trypsin enzyme which is there in the pancreatic juice, which is present in the inactive form as trypsinogen. This trypsinogen gets activated with the enzyme enterokinase. This is present in the intestine only. So enterokinase acts on trypsinogen to change it into active trypsin. Now, this active trypsin acts on the remaining proteins and polypeptides. Proteins and polypeptides on the trypsin acts to change them convert them into smaller peptides and amino acids. So, protein digestion starts in the stomach. Okay, the remaining part of the proteins will be acted on by trypsin, active form of trypsinogen, that is trypsin. Trypsin will act on the remaining proteins and polypeptides to change into smaller peptides and amino acids. Okay, another enzyme which is present in the pancreatic juice is trypsin. This trypsin will act on emulsified fat. Okay, so the fat first is emulsified by bile. Now, this emulsified fat will get more simplified by trypsin. Okay, from the pancreatic juice, this will change the emulsified in fat into fatty acids and glycerol. Okay, fatty acids and glycerol will change too. Now, this much digestion has taken place. See, now the sugar, the leftover starch has been converted to maltose. Then proteins and polypeptides, remaining proteins and polypeptides are changed into smaller peptides and amino acids. Emulsified fat has been digested to form, simplified to form fatty acids and glycerol. Then the intestinal juice acts on. Now, the intestinal juice, they are secreted by intestinal sucrose. The intestinal juice is secreted by the glandular cells, which is present in between the villi in the small intestine. Okay. Now, this contains what? Erepsin, maltase, lactase, sucrase, and lipase. Okay, so 5 types of enzymes are present in intestinal juice: erepsin, maltase, lactase, sucrase, and lipase. Now, all the actions of the enzymes are, in a nutshell, we are telling it that as peptides, the remaining peptides, smaller peptides, which the pancreatic juice is converted into, that peptides will be acted upon by erepsin. Okay, it is a peptidase. It will that is known as erepsin, that will act on peptides to form amino acid, end product of protein, that is amino acids. Okay, now the maltase will act on maltose to form it to glucose. Sucrase will act on sucrose to form glucose plus fructose. More simpler sugars. Okay, disaccharides will be also converted into monosaccharides, and that is simple sugars. Okay. Now, the lactase will act on milk sugars, that is lactose, to form glucose and galactose. Now, lipase will act on emulsified fat acids, the remaining emulsified fats to form fatty acids and glycerol. Fatty acids and glycerol are the end products of fat. So, lipase will act also on this intestinal lipase, that is acting on emulsified fats to form fatty acids and glycerol. Okay. Now, the absorption, the digestion part is over now. All food particles have been simplified more and more, and the simplest form of the food is ready for absorption. Okay. Now, the food has to be absorbed in the intestine. So, intestine has 2 functions, I told you in my previous videos. That is, one is digestion, the other is absorption. Now, we will discuss the absorption part. Now, the final product of the food which is ready now, is mainly absorbed in the intestine itself. Main absorption is generally in the intestine. The remaining part will be later on in the absorbed in the large intestine. Okay. Now, the amino acids and the simpler sugars. Okay, the amino acids and simple sugars, which are relatively small sized molecules, okay, they will be absorbed by the thin epithelium of the villi. We know the villi has single layer of epithelium, thin single layer of epithelium that will help in the absorption of the amino acids and the simple sugars. Amino acid is what? Proteins and product, and simple sugars are the end products of the starch. So the two products, that is protein and starch, that will be absorbed by the thin epithelium of the villi. Okay. Now, how are they absorbed? The epithelium of the villi, okay, when the cells are absorbing it, they will reach the blood capillaries. After reaching the epithelium of the villi, the food is absorbed by the cells, the epithelial cells, and they will transfer it to the villi blood capillaries. Okay. Now the food will reach into the blood circulation. Now the hepatic portal vein will carry this food to the liver. Okay, and then they will go to the main circulation, main blood circulation. Okay, so the first they will be absorbed by the epithelial cells of the villi. Then they will be transferred to the blood capillaries, carried to the veins, okay, hepatic portal vein, and then to the liver, and from there to the main blood circulation. Okay. Now the fat part. So we have discussed the protein and simple sugars absorption. Now we'll come to the fat part. How are they absorbed? The fatty acids and glycerol are absorbed by the lymphatic vessels or the lacteals. Okay. So the fat part is absorbed by the lymphatic vessels. The lymph vessels, they absorb the fat part of the food. Okay. The fatty acids and the glycerol are absorbed with the lymphatic vessels or the lacteals, and they are carried to the lymphatic system, and then finally they are transferred to the bloodstream. Now, after this absorption, protein, carbohydrate, and fats absorbed, still there are some food which are not digested, not absorbed. They will be carried to the large intestine by moving very slowly through the small intestine, taking about 4 hours. Okay. So you can see this picture of the bile duct, the pancreatic duct, the duodenum, okay, pancreas, the gallbladder. Okay. So this is the hepatic duct which carries the bile. After this, there is the liver here. Okay, above this hepatic duct, there is the liver from where the bile is carried by the hepatic duct. Either it is directly poured into the duodenum, or they can be stored in the gallbladder for some time. And after, when it is needed, they will be carried to the cystic duct and poured into the duodenum by the common bile duct. And this is the pancreas, the leaf-like structure. That's the pancreas, yellow, whitish yellow structure. Okay, this with the juice from the pancreas will be carried to by the pancreatic duct and poured into the same, through the same aperture into the duodenum. Okay, this is the duodenum part. Okay. So this much for today. We will carry on the discussion with the large intestine and assimilation of food, and even some tests of the food are there. That is carbohydrate test, and fatty acid test, okay, fat test, the protein test are also there. So we will discuss in our next video. Till then, please go through. If you have any doubts, please write in the comment box. Do subscribe if you have not subscribed yet, and do meditate before you start your day, because that will increase your concentration, give you interest in your studies, okay, and also you will get application. That is for sure. Okay. So please do meditate before you start your day. Okay. That will be a very good medicine for your memory power increase. Okay? So thank you. Bye bye.